states. We're not in Indiana or Illinois. We are here in the arid West talking about how we can make these principles work for us. So with that, I get the great pleasure of introducing Ms. Jill Clapperton. Jill Clapperton, PhD, is a principal scientist and owner of Rhizoterra. She is an internationally recognized educator on how to create, measure, and maintain healthy, productive soils that produce tasty, nutrient-dense food. So Jill, I'd like to welcome you to the stage.
Well, everyone, let me grab this mic because I am not tall enough to be standing behind this podium for very long. Okay. So I hope you all get just from this first picture what we're going to talk about today. We're going to talk about life in the soil. We're going to talk about who lives down there, what they do, and how we benefit from them. But we can't benefit from them until we actually create a habitat. So for some of you who heard how to create a habitat yesterday, we're going to review some of that, but mostly we're going to talk about this model. So first things first, soil biological activity is the driver of all of this. It drives, absolutely drives, the chemical nutrient cycling. It drives the physical structure. You cannot, under any circumstances, have soil health without having biological activity. I mean, you think about health. Well, health implies something alive and living. Your soil is alive. And as a farmer and a rancher and anybody who grows things, you have the ability to maintain that system. You have the privilege of maintaining that system. But you also have the privilege of changing it to do the kind of things that you want, and we'll talk more about that. Soil productivity. Oftentimes we're measuring soil productivity in terms of bushels. How much food do I grow? Well, I have this many bushels, I have this many tons, I have this many pounds. I would like to change that. If you have 50 bushel wheat, how good is it? Is it good seed? Is it good for me? Is it good for my livestock? Let's talk about that. What's in there that makes it nutritious? Quality seed is really, really important. Your seed, put in water, should grow a plant for 12 to 14 days without any nutrients. And if it can't, your seed is not good enough. And you need to talk about that. You need to think about that. You need to work with your seed producers to think about that. Environmental quality. Well, hey, if we're putting all the nutrients into the food, then that means our system isn't leaky, and we're not leaking anything out of our system, which means we're holding all of it. And when we hold all of it, it means that we put all of it in the food, and that means we're going to have better health for ourselves and better health for our animals. So this is all about using what we've got and using it to the fullest. So let's talk more about that, shall we? You have to build it first. You have to build the habitat first. And you can't build it if you do a lot of tillage because you're destroying the infrastructure. You're destroying these amazing cities that we stand on. And you're destroying all the energy that has gone into building that infrastructure. So we can't do that. If we want to take advantage of biological fertility and we want to have soil health, we absolutely positively have to have a no-till system or a minimal till system. We want to see earthworms in our soils. There are some soils in the world that do not have earthworms, but then they have Incutraids. And I'll show you what those are later. And they're very much like earthworms. But I want to see that chunky soil. I want to see it looking like crumbs. Let's have good structure. Let's make sure that we have infiltration. Make sure that every drop of water, we get all of it. These beautiful mites. These are microarthropods. These are mites. How many mites fit onto the head of the pin? About 14, give or take. So when you see this image, like the image at the front that I just showed, you can tell these things, that was magnified 50,000 times its size, just to give you that effect. And you know, I watch some of these sci-fi movies, and I see these guys on there. I'm like, oh, I know that. That's an oribaded mite. And there we go. And this is wonderful. This is the magic of Photoshop. So these things are all beautifully colored by Byron Lee, prepared lovingly by Derek Kaneshiro, who's my technician at Agriculture and Agri-Food Canada. And this takes a lot of work to do this kind of scanning electron microscopy. First of all, you have to mount this thing on a tiny pin, the tip of a pin, not the head of a pin, the tip of a pin. And then you have to dust it off. And then you have to coat it in platinum. And then you get to take pictures of it. And then you get to spend the time on Photoshop, lovingly making it beautiful, you know, doing the Vogue pose. This is the infrastructure. And this is an example of soil, continuous soil pore network. This is what we're striving for, and this is what we do not want to break down with tillage. The reason for that is a lot of the things that are really important in our soil for cycling nutrients cannot burrow. They need to zoom around in these channels, on water films, tiny water films, and they zoom around. And this is the only way we can have predator-prey relationships is by having this beautiful soil infrastructure. But once again, I said the biology drive all this. Well, they do. Earthworms make the most magnificent big tunnels that poop on the far side there. The little round ones, that's columbalin poop, another microarthropod. And it's creating the soil structure right next to the soil, eating up. When when you see the leaves, and all they are is lace, all they have is the midribs and stuff, everything is eaten out of them. Well, that's the columbella. They've eaten everything out, and then they left all their beautiful poop to create more soil structure, and then it creates more habitat. Again, we're creating another habitat because now we have soil microbes living around that poop, breaking it down some more. And you know what's beautiful about it? They're all living in and around the roots. So we've created this beautiful habitat. I'm going to keep going here. And when we do that, when we create soil structure, so soil structure is one of the most important characteristics of soil health. And you think, I told you biology was important. But a lot of people ask me, well, how do I measure it? Well, infiltration is one of the best ways to measure it. Because if your soils can have that water down, it means we have good soil structure. It means we have water-stable aggregates. It means that we have a habitat. So this is one of the nice ways that we can measure the habitat. And when we have nice soil structure, roots can move easily. And when roots don't need to use a lot of energy to move in the soil, they make lateral roots, which means that they go, oh, this is a luxurious habitat. I can just grow fat and really grow a whole lot of leaves and have a good time in here. And they don't use that energy, instead to force their way through and push through and try and find a way to get more nutrients. The other thing about it is when we have soil that has crumb structure, it binds to the roots better. And I'm going to talk a lot more about how it binds. And that means the plant has more access to the nutrients that are in those PEDs, in that structure. If it goes through the plates, it's trying to keep the soil close to it. But there's areas where the soil isn't bound to the root anymore, which means that it's struggling to get at some of the nutrients in the water. We don't want that. And when we've done tillage, and we've done tillage, factoid. You know, in the late 1800s in Illinois and the early 1900s in Canada, we talked about how much organic matter was lost. And with the first passive tillage, you lose more than 33% of your organic matter. But the good news is after 10 years of that, you started to only lose 3%. That's because you'd lost it all before. So what we want to do is start rebuilding, right? So this is all about rebuilding. I want you to look at these root structures. I love having glass plates and looking at roots going down. And you can create these nice little layers of compaction or light compaction, and you can see a little bit of compaction is not a bad thing. Look at the lateral roots that have formed because it has to try and explore a little bit. Now we have more root area. Now we have more area for microorganisms. Now the plant, you know what this is about? It's fitness. This is CrossFit, man. That root is hitting the CrossFit gym. Has to work a little bit harder. We do not want lazy plants. We want fit, strong plants that can grow in our environments. And grazing is no exception. We need all the water to go through. We don't want water running off our fields, and we can run off water in grazing. We can. And we don't want that either. Everybody thinks, well, I got some cover. Well, no, we actually need to have that infiltration. And part about that's diversity, because we need the soil structure, right? We need to have soil structure. So we're going to have good infiltration. So that means we're not running off. The other thing that happens, we saw that beautiful mite at the beginning. Well, mites are also intermediate hosts for tapeworms and a lot of little nasties for livestock. And when the ground gets too hot, we get too much bare soil, we don't have enough cover, up they come, and they start waving around like ticks on the top of the grass. The next thing you know, your animals are eating them, and you've got tapeworms. It all is connected. Everything we do is connected. We want the water. The rainfall simulators are wonderful tools for showing what's happening in our soils. I love the rainfall simulator. But I can have other rainfall simulators too. Whatever you need to do in the field to show this off, use it. Pop bottles or water bottles work great. Quick and easy demonstration of how soil infiltrates and how we get clean water. Because one of the things, we talk a lot about water cycle, when we're in the desert and we're in dry environments, and so for 16 years I was a Rhizosphere ecologist at the Lethbridge Research Center in southern Alberta. We are in a 9-inch rainfall zone. Some years I had 5 inches. I get this. But I also had real winter. Cold. So now I do double duty. I have a short growing season. So it's not that I'm not aware of this desert and the drought situation, but again, working to hold and have all the water, that's the key, which is why organic matter is so important to us. So now I'm just going to remind us again, okay, this model. So soil biota define soil health. Bacteria and fungi are your primary producers, and they can get out of hand. Bacteria can produce a whole new generation in 18 hours. You think they can't outcompete your plant for every tiny trace element? They can. Fungi, on the other hand, take about 48 hours to have a good run on the hyphae. But the beauty of fungi is that they hold nutrients for longer. So they kind of hold things in your system, whereas bacteria turn over very quickly. And then, of course, we have the predators, the protozoa, the nematodes, the amoebae, and we get into the microarthropods, which are all about breaking down all our organic matter. And then we get into the really big guys, the Enchytraeids, and Enchytraeids are still tiny, but then we get into the earthworms, the mega engineers. So fungi, if you start to ever see mushrooms in your field, you see mushrooms on your stalks like this, you know you've arrived. Because it means you're actually breaking things down. Fungi are absolutely critical for breaking down the cellulose. And in dry environments, they're particularly important because we tend to retain our residues a little bit longer, and we need to have them weakened. So first off, we need to have microarthropods drilling holes in them, and then we need to have the fungi going in and creating and eating up all the lignin. Trees do not decompose without fungi. People think about fungi and you think of all the fungal diseases. 95% of your fungi have nothing to do with disease at all. There's a very small proportion, and the beauty of them is that they are not fit, they are slow to get on the root. So if we can grow healthy, fast-growing plants, we can outcompete that. And if we manage our nutrients correctly, we can. And I'm always going to include little bits of, like, how can I measure that? Well, this is a new twist in an old test, the cotton strip test. So we used to use cotton strip tests a long time ago. I didn't use them. It was before me. But and they had tensiometers on either side. So they pull these cotton strips apart, and then you would measure the amount of decomposition. And that was an idea of how much fungi you had in your soil. Well, you do the same thing with underwear. And, you know, I was kind of tired of everybody using jockey shorts, men's jockey shorts. I was really like, well, really, we use women's underwear. Come on. Well, there's a little problem with women's underwear, and that is that it turns out that most of it is not really 100% cotton. There's this nylon mesh in it, and as somebody pointed out to me, it's like, well, that's okay. You could repurpose it. I was like, mycorrhizal fungi. They hold it all together. The glomalin that they leak out of their roots is this wonderful glue that glues the soil particles together and starts making something that is really water intolerant. So now we have real water stable aggregates. And fungi cast like this big silk net over all these beds, and we start to get really big soil structure. And now we really start to get this beautiful infiltration and more water in there and more air. And look at, when you look up close, you start to see that there's actually quite a lot of space in there, right? If you could Honey, I Shrunk the Kids and get right in there, you actually have a lot of room. So what I want you to think about is when you're thinking about things in the soil, think tiny. Think about, oh, I shrink down and I'm looking in there and I'm looking at something really small. Ever since the time I was a child, I loved looking in the microscope, and I still love looking in the microscope. I like to see what's underneath there, what's in there. And mycorrhizae are the ultimate carbon trader. That's what they do. They're trading photosynthate. They're trading mineral nutrients and water and for photosynthate. But mycorrhizae are even more important than that, and I'll show you the next slide. Mycorrhizae actually, endomycorrhizas actually live inside the plant. They're an endophyte. Not an endophyte like you're thinking, but they actually form a symbiosis. And the plant tolerates them completely. And they're totally dependent on the plant. They can't grow without a plant. They grow from spores, but they also grow from infected fragments that will live for a very long period of time, because the hyphae, as you can see, are really quite big and quite strong. They're living inside the root. They're conferring disease resistance, water resistance. They're actually transporting all these hard-to-get minerals, trace minerals in there. Now, why would they do that? They're not doing it because they're not selfish. They're completely selfish. They can't live without the host. So they're going to make darn sure that that host is as strong as it can be so that they can benefit from all that carbon. Now, everybody talks about sugars. When we talk about carbon, I would bet that most of you here think it's synonymous with sugar. Am I right? If I said carbon, is it sugar? It's amino acids, organic acids, lipids. So actually, mycorrhizae prefer amino acids and organic acids to carbohydrates. Carbohydrates third. Amino acids and organic acids come first. Now, why would that be? Because they also want the nitrogen. They want some of the trace elements that are part of those very complicated molecules. And if you're going to build more structure, what do you need? Amino acids. You need lipids. You need these other things. You need the building blocks in order to do this. We do too. Essential amino acids. Why do you want to eat essential free amino acids? Because you don't have to use energy to put them into proteins. You just combine them right away. Animals, bacteria, fungi, they're all the same. Everybody wants free amino acids. They want free lipids. They want all of that because it helps them build. Now, how many of you folks know about spurge? Spotted knapweed. Highly mycorrhizal. Beautiful example of mycorrhizal fungi in a root. That's knapweed. How do you think knapweed lives on really gravelly soils or invades things so well? It's because it has a competitive advantage over our plants. We don't breed, we breed plants with luxurious amounts of nutrients. They don't need mycorrhiza. So, are they really fit? No, because what happens if they don't get luxurious amounts of nutrients, if they live in tough climates, they can't compete. You know what? They can't compete without mycorrhiza, so that's a problem. Yellow color on the roots. This is a very old way to look at mycorrhiza, very old way. But it works brilliantly. So those bright yellow roots, what are they doing? Mining an old corn stalk. That mycorrhizal fungi is mining the old corn stalk for that new plant. So we want to use mycorrhizal fungi, and you can actually look at, it actually will be mining the old organic matter. How many of you think of algae? You might have seen this on the prairie. I've seen it in the desert here. Nostoc algae. It's a nitrogen fixer. And then when it gets really dry, it powders up into a beautiful source of nitrogen. You want to keep these algae. There's nothing wrong with algae. These are actinomyces. I've had people tell me that's mycorrhizal fungi, and I was like, no, really can't see mycorrhizal fungi in a root unless you're seeing that yellow color. So what you're looking at here is actinomyces. Actinomyces are wonderful things. They're kind of fungi and they're kind of bacteria. They produce a whole bunch of antibiotics. So when they live in and around the roots, they're producing a lot of antibiotics that keep a lot of the nasties at bay. So it allows the root to grow freely. And they're really good at decomposition. They give that soil that beautiful sort of sweet smell. Those are your actinomyces. You watch these guys, they're incredible predators on bacteria too. On other bacteria, I've watched them on a plate when I had some on a plate. And they just, like, the whole colony just comes over and engulfs something else and just eats it up. And you're like, wow, okay. Bacteria eating bacteria. Amoebae, protozoa. Protozoa, this is the wonderful work. This is a seminal paper by Elaine Ingham that really was her claim to fame really early on, was this one paper on how much protozoa contribute to nitrogen movement in our soils. Protozoa, but they do it because you have that beautiful infrastructure, right? They're moving around on those water films. They can't burrow. There's no burrowing for our protozoa. So they're moving around on that beautiful infrastructure, zooming around, and because they have good infrastructure, they have access to all these bacteria, right? So they can just gobble them all up. And in the meantime, they're also gobbling up, tiny mites and, new mites, some of the eggs of things, but they're just gobbling it up, concentrating the nutrients and pooping them out. And so then once again, we get nitrogen cycling, nutrient cycling. We create more habitat because now the bacteria are growing on that again. The whole thing works together. Nematodes are pretty much the same way. We think of nematodes primarily as plant pathogens. Let's not think about nematodes as plant pathogens so much anymore because once again, the majority of them are really good for our soils, really good predators. We love them. They also prey on insect larvae, amazingly. And corn, if it starts to get too much grazing on the root by corn borers and stuff, will actually send out what's called a sesquiterpene. And that terpene will actually attract more nematodes that are going to prey on that insect larva. Just because I'm rooted in the ground as a plant doesn't mean that I'm not on offense, right? I have to be because I'm stuck. I can't run away. So I'm going to be offensive and I'm going to have good defense as well. And so plants are not just passive and they're not just rooted in your soil. There is a big interaction going on in there, and these things are critical. And that little band around that nematode is a fungus called a snare fungus that is actually trapping nematodes and eating them up. It's a pretty rough world down there. And then look at this guy. This is the vogue mite. This is an orbatid mite. This has been on the cover of a lot of magazines and is in the new Encyclopedia Britannica. Look at the mouth parts on that guy. This is a tank going through everything in its way, chewing it up. Now you'll notice there's no eyes. No eyes on any of these things. You don't need eyes. You're in the soil. You're not going to have a lot of light down there. But you'll notice that there's a lot of hairs. So how do you know which is predator and which is prey? How do you know who the good guys are and the bad guys are? Vibration. Those tiny hairs on there set off a vibration, and they sense the vibration with their antennae. And we know that because of the wonderful nanotechnology that we have right now. These are Columbella springtails. See the kangaroo tail on there. They're those little guys that hop away. You pick up the litter at the top, and then you hold it up, and then all of a sudden all these things, just little tiny things, spring away. Those are springtails. Now, these are actually related to insects. They have actually an eye cluster, and I think I can point to it with this. Maybe not. There's an eye cluster up the front. There's about 3 of them there. And that eye cluster, they actually move up and down. So they're actually living in the litter layer more or less. And they're really important because they eat out the stuff in between all the lignin. And by eating out all that soft stuff, and they also poke some holes in the lignin, which allow the fungi to go in. And you know the best thing about that is that mites and Columbella love organic matter that is colonized by fungi, self-perpetuating. In science, we call it metabiosis. So one thing is creating a habitat for something else, and then something else, and then something else, and you have this beautiful cascading effect. I promised an Entrada. A lot of people see these and they think that they're potworms or nematodes, and then they're like, oh, that's bad. No. If you actually look at them with a glass, like any kind of a magnifying glass, you'll see that they're segmented. And they're just actually microscopic earthworms. And they're related to earthworms. And once again, they're about concentrating nutrients in and around the roots. And really, really important. And I put that with an earthworm because I wanted you to see the size difference. And then, you know, I mean, we're getting close to lunch with a little bit of sex in there. Earthworms. Earthworms are what we call ecosystem engineers. They make megatunnels, and they really strongly affect the infiltration and aeration of your soil when you have them. The night crawlers that people get in their lawns are very rarely found in the field because they like an environment that is really luxurious. They need water. They need to be pampered. They're long-lived. Earthworms, night crawlers, will live up to 8 to 10 years. And they'll only mate when they find another earthworm. So that what they do is they keep their tail in their burrow, and then they extend their bodies out in a radius around that burrow and hope that there's somebody else out there that they can mate with. And if they don't find them, then off they go and wait for the next time. And so why do earthworms in a rainstorm, why do you see them all out? Anybody, venture a guess, otherwise I'm going to have... Exactly. So otherwise the surface is too dry for them and they can't move around. There's another reason. They sunburn really easily. So in a rainstorm, the sun is diffuse, and so they don't easily burn. So not only can they move fast, but they don't get burned, and the birds probably aren't out preying on them, because when we do worm watch with kids in the classroom, first thing they do is show a robin or a bird preying on earthworms. That's the first thing that they draw. And we just don't have as many birds out in the wet. So what happens is it's the perfect time to go on a walkabout and see if you can find somebody to mate with. Now, if you're an earth worker and not one of these big night crawlers, then you don't need to find somebody else. You can self, because earthworms are hermaphrodites. And so they can actually self. They don't necessarily need to find somebody, but these guys are earth workers, and they found each other in a rainstorm and had fun. All right, how do we create healthy soils? Well, let's talk about that. There's only one way. You created the habitat now. You're no-till or you're minimized your tillage. You're not destroying your habitat all the time. Now you have to have plants because no-till doesn't work without crop rotation. It doesn't work without the plants. It doesn't work at all. I mean, people say, oh, I remember, because I came from the Pacific Northwest. And the first time I learned about no-till was with Dwayne Beck and Roger Veseth, and it was the first time I'd given a talk at a no-till conference. And they said, do you think anybody will care about this stuff? And I said, I don't know. I care. And I was prepared to talk to one person in the audience. It was okay. The rhizosphere is the most important. I can't emphasize this enough. It's the most biologically active, the most important area in your soil. It is what generates soils. Soils do not generate anything without plants because plants feed everything that's below ground. It's not just about these old residues that are on the top. No, it's about the roots. How do we get carbon into the soil? Roots. You don't get it from dribbling from the top down. No, you get it from planting roots. And the roots leak. And they leak a lot of carbon substances, right? Not just sugars, they leak a lot of amino acids, they leak organic acids, they leak lipids, they leak a lot of things. And then that feeds, and so what you have is this partnership. The plant roots associated with the soils and the soil organisms. And every different plant you put in the ground, every different one, creates its own signature of root exudates that attract what it wants, what it needs to be healthy. Remember, that's offense now. Hey, I need rhizobium. I need mycorrhiza. I need other things. How many of you knew that legumes actually have two partners? Not just rhizobium, not just a nitrogen fixer. They can't actually fix nitrogen without mycorrhiza. Why? Because they need zinc and iron. You can't make a heme, that pink-red color that shows you that your nitrogen nodule is working, without iron and zinc. So you need the mycorrhiza. So they're actually a tripartite symbiosis. Roots. Lots and lots of roots. Let's grow companions between our crops. Let's grow intercropping. Let's get roots, 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 because roots are going to make carbon in our soils. Roots leak things one way, but they also leak nutrients. Unfortunately, sometimes they have to leak nutrients at the same time. That's why I talked about seeds. Sometimes early on in a plant's life, it will actually have to leak nutrients because you don't have the nutrients in the soil to feed that plant. So it actually will leak nutrients in order to get it. It will leak, and it might leak them even to just change the osmotic potential so it can get water. We never want that with our seedlings. We never want them to be sharing with the soil, ever. But that doesn't mean we have to overdo it, because if we overdo it, then we create an osmotic gradient again, where maybe the plant has to leak because we got too much fertilizer, and it needs to sort of like dilute it down so it can actually survive. So this is a fine balance, and like everything in life, it's a balance. It's all about the balance. And then you have growth-promoting substances. And that's what the plant's looking for. It's looking for the bacteria that actually exude plant growth-promoting substances. Auxins, gibberellins, cytokinins. All of these things will actually help, but mostly auxins. And other things, other complicated molecules, enzymes and whatnot, that allow it to actually get more nutrients. And then you've got your pathogens. And remember I said they're really lousy competitors? They produce plant-inhibiting compounds because they're trying to slow the root down so they can get on. So we don't want to slow roots down. That's why we don't want compaction. That's why we don't want others, because we can't afford to have the roots slowing down. We need them to go fast so that they can't, so pathogens cannot get on. So which one do you want? Mycorrhizosphere or just a plain old rhizosphere? We want the mycorrhizosphere. We want to trap water. And remember I told you that the mycorrhiza are all about preserving the plant life because they need it. Now, the mycorrhiza do something really cool too. Is they know that they're a drain on the plant. They know. They can be a drain on the plant at times. So when the plant changes over to forming a seed, mycorrhiza are already finished and they formed all their spores and they are finished. They don't need anything more, and then everything, all the goodies go up into the seed. They are totally in sync with that. They're trapping a lot of moisture, they're trapping a lot of bacteria, and they also, the mycorrhiza, because they change the root exudate pattern and the composition of the root exudates, that rhizosphere is full of Bacillus thuringiensis, has actinomyces, has everything about protecting the plant, right? Because once again, the mycorrhiza is selfish. Needs the plant to be protected, so it's making sure that mycorrhizosphere is very protected. I love this little slide from DSV Seed. They did such a good job on this. And then what the goodies? I have to keep pressing it. There we go. So what are we going to do? We're going to grow diversity because we're going to fill our carbon profile. We don't want a monoculture because we just get everything in one spot, and that's easy for competition. We need to actually vary some things. And we had talk on the first day about calcareous soils and about high pH. Well, look at those roots. First one's faba bean. That is a very low pH that it is exuding in great tea out of its roots. I love faba beans. Not only do they have big fat roots and little tiny shoots at the beginning, but those roots just pump nitrogen, have beautiful nodules, and they do this. Then in the middle we have soybean, and you could see that the soybean is also a bit on the acidic side, but a little bit more basic, right? And then you can see corn roots are actually pretty on the neutral, more basic side. So why would we want to grow corn with a legume in between if we're in calcareous soils? They're going to feed each other. They form connections. The mycorrhiza form connection between the corn and the legume. And yeah, they form connections. In the Pacific Northwest, in Oregon, they have this beautiful project where they actually did carbon-14 labeling, and they labeled one tree. And then they watched it go for 25 kilometers through the forest. Because it's networked. The mycorrhiza network the entire row. And if you have stuff between the rows, then they've networked your entire field. So how cool is that? And that's why when we have mixtures and we have diversity, we aren't as subject to drought. We are going to use more of it. Water and exudates. Jimmy showed a really nice picture of that the first day. But look at that forming, beating up on those roots. It's beautiful. So let's talk about that whole sharing thing. This is a monoculture of common bench. This is the drawdown, the water drawdown. You can see the water drawdown at all 3 depths. Now we put in a mix with 12 different species, and oh my gosh, there's no drawdown. What the heck happened? How does 2 and 2 not equal 4? Instead, it's equaling 8 or 10 or 12. How does that work? Well, I'm not going to tell you we know, but we know that that networking thing is really, really, really important. And that plants thrive in community. If you don't have a community around you, how well do you really do? How many people are suffering from, and I don't mean just in this room, but how many people are suffering from mental illness now because of the isolation because of COVID? We do not do, nothing in nature does well in isolation. We're all connected. So let's grow things together. Let's max out on photosynthesis. Faba beans and crimson clover between the rows of sunflowers on the eastern slopes of the Rocky Mountains outside of Claresholm, Alberta. That's southern Alberta, folks. Big winds, dry temperatures in the summer. That looks good. Dry land on 8 inches of water. Now, everybody thinks, okay, well, I've got to have that cover crop. I've got to have the dead residues because that's what's feeding my plant. No, actually, plants form connections, and they feed directly. So you can get phosphorus, nitrogen, and trace elements all through the network. It's like having your own intranet. Getting a nitro shot in the season. This is interseeding. This is Derek Axton from Minton, Saskatchewan. Jeremy Wilson from just south in North Dakota does the same thing. And we're doing intercropping. Intercropping in dry situations is probably a saving grace, and I suggest you check out Axton's. Desi chickpeas and wheat. This is my intercrop. It's not an intercrop so much because with the wheat I won't get a chickpea crop. But I use the Desi chickpeas. They're cheaper. They're also more wild type, which means that they actually take up more phosphorus and feed my wheat plants a little bit more phosphorus. And you can't use a herbicide. Everybody goes, well, if I do this, where's the herbicide? Well, that's the whole point. I don't want to use it. So, and I'm feeding my plants directly. And no, it doesn't compete. When I harvested that, when it goes to the combine, I don't get a lot of green. I don't get greening on my seeds, so I still get the premium. Well, there is no premium. I just don't get docked. Pasture stitching or oversowing pastures. Why not? Increase the diversity in that ryegrass. Increase the diversity in that brome grass. Let's start getting some diversity in there. There's real ways to do this, and there's not enough time this morning to talk about how exactly to do that. But it's possible, and we do it all the time. We're managing the soil for plant and animal health with the diversity. So now, you know, my girls shine. Their eyes are bright, their coats are beautiful, and that's what we all want. We want our girls to shine, and we want our girls to have beautiful calves, and we want them all to have calves. I want 95% of my calf crop. And if I could have 100%, I'd take that for sure. And why? And animal grazing is so important to the soil. Okay, this is phospholipid fatty acids, which are actually about looking at the total living biomass, because phospholipids make up the membrane of living cells. And when a cell dies, the phospholipids are the first thing to get eaten up. So that's why phospholipid fatty acids are so important. So clipping is a C. Then we have a grazing exclosure, a clipping exclosure. Then we have grazed and we have ungrazed. So we stop the grazing and then just let it grow. And you know, when you look at that purple bar, which is the total number of organisms in that soil, look at that happens when that grazed. Way better than clipping, way better than the exclosure, which never had any grazing. And then when we have grazed and then we stopped it, which is ungrazed, then we actually have a really nice example too. So what's really interesting there is we started to look at, I was doing that with Ben Bartlett, who's the extension veterinarian from Michigan State. And cows wrap their tongue around and then they pull. And when they pull, the roots go. And when they do that, they leak. And when they leak, we get more things growing around the root. And you know what? Beautiful. It happens within 2 days. So how important is having livestock on the land? Very, very important. And look at the diversity here on soil structure. Remember I said we talk about biology and how important it is for soil structure? This is a dairy farm I started working with. And so what we did is we just took a bunch of seed scrapings, threw them in the fields, you just broadcast them. And then we had ryegrass, just plain ryegrass. And the first time I did this, I'll tell you, I was out with a dairy group, and I had no idea if this had worked or not. I didn't. So we started with the pure ryegrass, and we had to get the heaviest, biggest guy to pound the shovel in because we couldn't get it in otherwise. And then we asked the heaviest, biggest guy to do that in the rows that had all the seed. And he fell off the shovel because it just went straight in. And we all stood there and went. And then we flipped it over, and look at that. 6 months later, we had changed. You can change very quickly if you want to. Okay, so now we have multi-species covers. And then we have a yellow mustard. Yellow mustard is great, and I've seeded pastures with mustard where I've had to deal with nematodes or some other things, and it works really, really well. But again, it's just one species, and I'm not changing things too much. And then look at that. Which one do you want? Multi-species. Now I got a habitat. Now I got good soil structure. Now I'm cooking with gas. I want you to plant with purpose. This is a trademark of Rhizoterra. I want you to plant with purpose. I plant with purpose. If I want to smother weeds, I plant a certain cover crop or a companion. If I want to feed my cash crop, I plant with a purpose. If I want to mine calcium and magnesium and phosphorus, I plant with purpose. I plant a lot of buckwheat and I plant other species with it. I plant with purpose, and I look at the genetics and I look for things like this and go, hey, I have a sandy soil. Why wouldn't I, this one on your right-hand side, why wouldn't I plant Phacelia if I have a sandy soil? Phacelia has these most beautiful fine roots, and it just aggregates the soil so beautifully, and it's mycorrhizal, and it helps pollinators. Ooh, why wouldn't you want that one? And I'm not going to leave out potatoes and sugar beets out of here. Why can't we grow things between the furrows? Why not? When we're harvesting onions, we have to do tillage. We have to lift the onions out. I work with one of the largest organic onion growers in the Pacific Northwest, Anderson Organics. And when we're lifting the onions out, we get halfway across the field, the drill comes in the other side. You got irrigation. Why wouldn't you throw a little water on after you harvest the onions and get that going and keep your soil covered? Why wouldn't you? And with here, it actually affects the yields of the potatoes and the sugar beets as well. That's the beauty of it, is that we'll actually, when we get it right, it didn't matter actually what we planted in between the rows. We're always better than the control, but some of them were amazing, like we did 14 tons of potatoes. Micronutrients are really important for plants if we want them to be strong and to have a really healthy immune system. No different from us, and this COVID has really brought that home to us, is plants need trace elements too. They need the zinc. They need the manganese. They need copper. They need all these trace elements in order to make these really complicated molecules. And when they get too much nitrogen, they can't do it. So it's about finding that balance. We need nitrogen to grow proteins and do things, but we don't need too much. So we need to find balance. But I promised I'd talk about food quality a little bit. So this is Fred Fleming, who's the co-founder of Shepherd's Grain, which is sort of a known flour company in the Pacific Northwest. So they're a company that came out of the co-op for the Pacific Northwest growers. And they're all no-till. They have to be no-till to be in that group. We started looking at cover crops. Okay, we chose 15 different mixes, and we were just beginning. And so we didn't know. We thought, well, 3 might be a little wild. But we did 2, and we were trying to replace a fallow because, honestly, fallow is just starvation for microorganisms. And so it was like, God, we can't have that. So, and our goal was to produce enough nitrogen and to have enough residue and have all these organic nutrients so the plants could take them up. And this was in Lethbridge, so this is dry land. And what happened there was, well, number 9, which when we will go back to that slide so everybody can see. Number 9 was subclover, sorghum Sudan, and buckwheat. And you can see it was 1 to 1. Okay, so for every pound of biomass, I grew a pound of nitrogen. Well, that seemed like a good idea. And then we grew wheat, CDC teal afterwards. And we looked at, well, what happens to the subsequent thing? Because what I tell you, no-till doesn't work without crop rotation. So what do we do? Well, cool. Faba beans, peas, and oats were the winner. But, we did okay on dry land in that year. What did I say? We had 8 inches of water, and I still was doing 65 bushel. I'm not going to complain. It's good wheat. And we started doing this, organic. So the second one, rotation 2, is just wheat fallow. And when they say organics can be rubbish, that is rubbish. That is rubbish wheat. It's not, I wouldn't even feed it to livestock. It doesn't have enough nutrition. That stuff isn't good for you. But now, let's look at when we put sweet clover with it in the and we do some other things. Oh, yeah, cool. So the low input there, actually I got confused. The low input, which is almost like regenerative, but it's a wheat fallow, so that's not very regenerative. You can see that wheat is rubbish and the organics is way better. But then when I even it up and I start to add more crops into the rotation, everything becomes very even. But then something really wild happens. In 5 and 6, I added a forage in the middle. So I did a cover crop. And all of a sudden, my nutrients went off the chart in my wheat. And I was like, well, I wonder what happened there. And then we started to understand. And you know what? In 1910, the Confederation, that means I'm up. Come on, turn off. That means that with the Confederation of Canada, because we didn't have Newfoundland at that time, and a couple of other provinces, they reported that, if you had a mixed forage in the middle of your rotation, that you actually got a 10 to 15% yield in your wheat. There was no fertilizer in 1910. They knew this already. This is old stuff. So we started looking at varieties and like wheat, and okay, so there are other things to do with varieties, and I started to work with Bob Quinn in Big Sandy, Montana, which is another drought-stricken area, and you can see that the variation is huge. And it wasn't just old varieties, it was new varieties. Some of the new varieties were great, some of the old varieties were great. Why don't we blend? And that's what Shepherd's Grain does. They blend up a bunch of different wheats to get the nutrient density that they want and the baking quality they want. So I want you to remember that when you stand on your ground, you are standing on the rooftop of another world. And you have the privilege of being able to manage that whole other world. And what you do on your farm and your ranch will affect what they do for you. And why aren't we taking advantage of soil biological fertility? Instead of always tilling Mother Nature into submission, why don't we work with her? Oh, that's a novel idea. If you want to know more, this is a shameless plug for my network. But all the talks I've given are on there, and they're all in the library, and you can watch them at your leisure. Thank you so much for coming out today. I really appreciate your attention, I really do. And we even have time for questions. So, I'll open it up to questions. Thank you. Yes?
Do you help me understand a little bit better when you talked about legumes and their rhizobacterium connection, that it's actually not affected without a mycorrhizal connection also, which means that the legumes don't work with mycorrhiza, right?
Oh no, they work with mycorrhiza. The only legume that does not work with mycorrhizal fungi are lupins. Lupins. And lupins fix nitrogen in a very different way as well. And so does rooibos tea and some of the tea tree plants. They're still legumes, but they fix nitrogen in a different pathway than what you think of as a clover or alfalfa. But all what you're thinking of a legume actually are a tripartite symbiosis. They require mycorrhiza as well as the rhizobium.
So alfalfa will create that.
Yes, it will. And alfalfa loves corn, and corn loves alfalfa. They love each other, you know, they're just a marriage made in heaven. And they're like, oh, let's share mycorrhiza, let's share resources. Let me give you some nitrogen. Oh, why don't I give you some of this? You know, and you know what? A lot of farmers early on knew that, right? I mean, they knew, in Minnesota, they talk about, you know, you can, you know, have the Sven and Oli jokes if you want, but they were like, oh yeah, when I plant the alfalfa, the corn goes really, really well. And so they loved it, and they knew it. They knew that you needed to grow them together. So they always grew alfalfa, and then they grew corn right after. And a warm season after an alfalfa crop, like if you terminate an alfalfa crop, A warm season after is the only thing you want to try. Do not grow wheat. Do not grow something with a puny little root system that doesn't like a whole lot of activity. Grow a warm season grass. Grow a millet, grow corn, grow something like that. Yes? I can. So saprophytic fungi are more about decomposition and breaking things down. So what they're going to do is they're going to break things down, and they're free living, totally free living. They just are looking for residues, and they're just going after them, and they're breaking them down. Mycorrhizal fungi cannot live without a host. So they cannot live without a living plant near them. And then they can't live without actually colonizing that plant and forming that symbiotic relationship so that they can survive, because they can't even reproduce unless they're actually colonized a host.
So are they there before the plant grows?
They are. And in fact, we know that mycorrhizae co-evolved with the evolution of plants. So they have been connected together since the beginning of time.
So would you say then that they're the islands, so they're the most important living?
They are, but here's another thing though. There are some plants that do not form mycorrhizal associations, and brassicas are one of them. But brassicas have another role, a really, really important role. Brassicas are Red Bull for your microarthropods. And I mean Red Bull. You know, there's that animated movie, Over the Fence, and the squirrel drinks a Red Bull and goes crazy. That is your microarthropods eating brassica. Yeah, no, just the brassica, even the brassica leaves, like anything to do with brassicas. Oh man, earthworms too, they love it. Not sure exactly why. I can't tell you why. I know that the carbon to nitrogen ratio is a bit higher in that residue. And so I know that the quality of organic matter is really important, but it stimulates activity of all those animals like you cannot believe. So that's why oftentimes we like growing things like mustards or canola with peas and whatnot, because then we have both, right? So now I'm stimulating the heck out of all those microarthropods, and I got the mycorrhiza to boot. Woo! It's a twofer. Well, you know, I think that's exactly right. And I'm glad that you brought up that preg check your cows. I would tell you that we've been experimenting with that on some bison that we've been really struggling with because of, you know, just losing fertility a little bit in there. And when they start eating some of those covers, just like the cattle, I see with the dairy, when I stitch pastures like that on a dairy, all of a sudden I go on this one dairy from 70% calving to 85 in 1 year. You're darn right you need to pay attention to that. Anybody else? Yeah, and then I'll come here. Saliva is really important, and cattle produce liters. Okay, there's my Canadian and my metric coming out, my scientist in me. It's like, let's say gallon, okay? But they produce a lot of saliva, and that saliva is really important because it has different enzymes in it. And so those enzymes are also helping in the soil as well. So yes, saliva is also really important because you see, with that same thing, like you see the tongue wrapping around and pulling, you know, the saliva dripping out. Yeah, it all has a role. So it's not just about urine and dung, it's also about the saliva and that act of pulling. Just a minute, I got one over here. Yeah, and then okay.
And two, it
Okay, it severs the network. So that's the thing, is the network, if we leave the network pretty much intact, then we're not severing the network, and it's easy. And if we do, even with a disc drill, we go in there, we're severing some of those connections, but we're rebuilding them very quickly. And you can, I really believe this, I really believe that spores are incredibly resistant and resilient and to weather conditions. They're resting stages. So we can inoculate in soils that are really, like if you actually do the test. So here's a little trick, everyone. You want to know about mycorrhizal fungi. Take your soil, put it in a tray, plant some onions. Onions have beautiful straight roots, and they form that yellow color almost right away. But the yellow color is very sensitive to light, very sensitive to light. So you have to wash them off really fast. So you grow it and put a little sand with your soil, just to make it easier to pull them out. And then dip them really fast and look for the yellow color, and then evaluate the percentage of yellow color. Now you have to have reps. So I want you to have at least 12 different seeds in there and seedlings, and then I want you to evaluate the yellow color. And then do that after you've changed your system a little bit and see if you get more yellow color. The percentage of yellow color, and that's actually up on my site. It tells you about how to do this old test and how to look at the percentage of the yellow roots. And it's the fastest way to look at that, because otherwise you have to stain and do all these other things, and you know, that's just really tedious. So, but this is really fast. And it's a really quick way to test if you're doing mycorrhiza. I like to say if you build it, they'll come, because they will. But if I need to speed it up, I look for inoculants. If you're going to buy an inoculant, you need to buy one that has more than one species. And I want it, if you have more than 6, that's a good thing too. So you want to have that. But you don't need it, and you don't need it every year. And the other thing is long time ago I published the Dilution Experiment, but everybody, I mean, as a salesperson, you always want to sell the most that you can. I mean, and I'm not dishing anybody or anything like that, because of course you do. But the problem is that a lot of times the diluted effect is way more efficient. And so you have to remember, if I put a small root into a whole bunch of hungry mycorrhiza, what are they going to do? They're going to hit that plant hard, and they're going to suck a lot of photosynthate out, which is going to slow it down. Now, I dilute it a little bit, and I maybe do a quarter of what they say. I still get the effect, but I don't drag on my plants. That's why I love granulated inoculants for legumes, because I put it underneath them. I don't coat them in it, because the legumes, the inoculants do the same thing. That is a bit of a carbon drain on the plant. So if I don't drain them right away, I give them a chance to kind of establish and stuff, right on. Okay? Okay, then we had a question here. Moles and gophers. Well, you know, it's kind of like mosquitoes. I like to think that they all have a place. You know, you got to think it's a big tunnel, like a lot of water rushing down into the ground. They're great for, you know, if you allow predators. And on the ranch, I allow predators. So I am not a big fan of hunting predators if I don't need to. And I also don't hunt snakes. I don't like snakes, but I don't hunt them either, because I know how good they are at getting rid of moles and voles and gophers. So I leave them. I know I don't see a whole lot of mice under the house anymore, because I have 2 big bull snakes that live under there. And they do a really good job on rodent control. And that's the thing is, I stop people, even in Montana, when I was living in Montana, I was like, you know, we can't kill snakes. We have beautiful rubber boas, and you leave them alone because they eat a lot of rodents. And people were complaining about all the voles in their garden, but did they see a snake? No, probably not. But once you start seeing a few snakes, there's no mice and rodents. Okay, Tony. You have to be really careful with wheat in a cover crop, because if you get a really good cover crop and you have a lot of biological activity in that cover crop, wheat is not as strong, can host a lot of that activity the next year. And you have to remember that all that activity from your cover crop is waiting to get on a new root, a new living root. It's like, yeah, look, there's a new root in there, and then you get this puny little wheat root that goes, ha, get away from me. And starts, and then it can't, and then all these bacteria are like, but we want on. And then they start gassing each other, and then they produce hydrogen cyanide gas and really nasty things, and the plant root goes, oh my gosh, really get away from me now. And everything slows down. So that's why with cover crops, when we're managing cover crops, we need to try and get warm seasons or get broadleaves in after. No, moisture, this comes back again to that slide of where I showed you, you get multiple species in there, and they survive moisture, drought, way better than they do as a monoculture. Yeah. Sure. And and by the way, this afternoon, I am going to talk about how to make the transition. But yeah, it's really important because if I just switch to no-till, what have I done here? I have started to create a stable habitat. I've started to create a thriving plant, a thriving community, below ground community, a thriving one, right? It's like growing a town. So I start with, maybe a few houses, and now I start to grow into a big city. And I start growing and growing and growing. Well, okay, I need resources for that. I need diversity of resources. I need a diversity of plant material in order to feed that growing below ground system. It's like a weaning thing. So now what you are doing is you are weaning yourself, and you're weaning all your plants off of this dead dirt situation into this living system. And they're all struggling. Everything in the soil that's starting to grow is also struggling. And so when we put diversity in, we start to have a diversity of organic matter. We start to feed them differently. We start to build more soil services. And then when we put crop rotation on top of that, and if we can use intercropping or we can use a cover crop, then we start to build the soil really fast, and we build the infrastructure faster. The last thing, though, and I will say this afternoon, is that when you start into no-till, you need to use more fertilizer. You do. Don't let anybody tell you, I can walk away from it. No, that's garbage. You cannot. Because you're feeding this whole community. I'm not feeding 5 people. I'm feeding hundreds, thousands, millions now. And so if I'm feeding them with a cover crop, fine. I don't need to worry about the fertilizer so much. But if I'm trying to feed them with a monoculture, oh man, that is a disaster. And it has been a disaster. And people have gone, wow, that was a dumb thing. Why did I ever try that? And that's because they weren't coached about there's a way to do this. It's like there's a way to transition to organics, that you can be successful, but you have to take these steps. And it's like a recipe. It really is. I mean, it's not prescription of the plants, but it's a recipe of diversity and the steps to do it. Why don't you, no, I wasn't, but why don't I just answer that right now? All right. I like whole numbers, so I like bacterial to fungal ratios because I don't like to see like 0.17 and things like that. I like whole numbers, so I just do bacterial to fungal ratios. I like them, and this goes back to the fungal question because I realized right now I didn't answer your full question, but I'm about to now. I think that if I have a super fungal dominated, if I start to see fungi, if I start to see numbers that are like 3s and 4s and 2s, which means I have a whole lot more fungi, and yes, that's right. I have a whole lot more fungi. That's a bad thing because I don't want a bacterial dominated system and I don't want a fungal If I've got a fungal system, that means my whole system is into decomposition. And I'm struggling now because now I'm probably breaking things down so fast. I got nothing on the ground. I got bare soil. I'm in trouble. I like it around that 5 mark, and I even like it 6. 6 meaning I have more bacteria than I have fungi. Now you have to understand the PLFA, we have a lot of problems with the biomarkers for mycorrhiza. So we don't represent the full fungal community very well. So it would be higher, yeah. I mean, we don't have a good biomarker for, we might have a new biomarker for mycorrhizal fungi, but we don't have a really good biomarker for mycorrhizal fungi. So we probably underestimate the fungi anyways. And it's a lot easier, the phospholipids for bacteria are a lot easier to find than they are for fungi. And now some of that's the way we do the chemical extraction. I'm not going to get into that, but we underestimate fungi in those numbers for sure. Protozoa, I really like the PLFA for protozoa, but once again, you're looking for triple bonds, double bonds in those phospholipids. And if you don't do a good job with the extraction, then you miss out on those, and we probably underestimate. What I do like, and if you look at a test for phospholipids, I do like seeing that precursor to end product ratio. They should show that, a precursor to end product ratio. And the reason for that is if I have a lot of precursor, it means my system is in a building phase. If I have a lot of end product, I'm kind of at the end. And I might be really stressed out because I'm at the end. So that's an idea of looking at your microbial community and going, okay, they're a little bit stressed, maybe this and that. And you know, so there are ways to look at that. But I don't want my systems totally fungal dominated. I really don't. If I can separate mycorrhiza, fine. But what's a fungal dominated community? One that's high in decomposition. I need residues. I don't want them all eaten. I need some armor. So I'm still going to look for that 5, 6, the break even or slightly higher in bacteria with still enough fungi to count. But I start getting into 7 and 8s and I'm like, oh, I totally need more fungi in there. Oh, absolutely. Yeah.
So when you start seeing that come down and your fungi coming up and reaching closer to
That equilibrium point, yeah.
so that's where you're saying,
I like that. And I don't even need to do the test. I start seeing mushrooms in the field. Like we're starting to see mushrooms now in our onion fields on the residue from our covers. We're like, huh, we got it. We're doing something right. Or does that mean we're going to stop and we're not going to tweak? No, we're going to keep tweaking. Because what farmer can't resist the urge to tweak a little bit more? Right?
can I just a little bit,
Yeah. They're having a hard time hearing you. You're muffled. Wait. Let's take a pause on that one right now, because I'm going to have to politely disagree that they take them up in only in inorganic form. And I don't want to get into that right now, because amino acids are an important source of nitrogen and phosphorus and some trace elements for plants. So we'll leave it at that one. Fungi are very good at holding the nutrients very well. Yes, go ahead. Mm-hmm. They should capture some roots with it, old roots with it too, because a lot of the old roots will have mycorrhizal structures in them. And that's a very important source of inoculant, which is another reason why when we have colonized roots and we build up all that organic matter, we're also building up our mycorrhizal inoculant. No, they can capture the bulk soil, not so much, because remember that if you're sampling between the rows, so when I sample soils, I sample right beside the plant. And if I'm sampling for PLFAs, I see the word hat. If I'm sampling for PLFAs, then I'm actually going to sample beside the plant and I'm going to take roots with it. Yeah. My own? Ward is one of the few, Ward Laboratories in Nebraska is one of the few labs that do PLFAs, and I taught them to do PLFAs. So, you know, there's a lot of other labs that do a great job. I send a lot of stuff to Midwest too. I've sent things to a lot of different labs. I mean, if you're interested in pesticides, then there's a lab in Ohio run by John Fagan who does a great job on things like that. I mean, you need to look around. And here's the one thing. Laboratories don't want to necessarily know I do this, but I always send a sample that I know exactly what's in there. I always send a sample in to test them. And if it doesn't come out the same, hey, you guys need to reanalyze this. It should look like this. And so dry out some of your soils, get them analyzed, send it to 2 or 3 labs, get the analysis on that one soil, hold it as a reference sample, and send a little bit of it in with every soil test you take. No, I haven't. No, I haven't. Probably should since I was in Washington, but there's a lot of labs in Washington, I can tell you that. Okay. There are labs that do water extraction, like use water to extract things, yeah. I do, and the problem with that is, and I don't have that slide up, but what happens is when you get a lot of chlorides and you get sodiums and stuff, you know how you get that browning on the leaves, on the margins? What that is the potassium pushing all that nasty stuff out to the edge so it can reassess its osmotic potential. So, you know, it's the dilution effect. Don't want too many ions and things like that. So it pushes it, so what's happening? You become potassium limited, right? Because all the potassium is being used to push all the nasty stuff out to the margins. And when the margins die off, then you got potassium again. So it creates some real issues with it. There are some really nice polymers now that they're using that will actually bind onto some of the chlorides and the sodiums and whatnot that in your irrigation water, which means that it's not as biologically active, and which means that we don't have some of the same issues with that. So I don't recognize you, Jerry, without your big hat on. Yeah, that's your signature. Come on, that big hat. Okay. Yeah, go ahead.
You mentioned pesticides, that type of stuff. What is glyphosate, Ivermectin, all that to do with biology?
Well, Ivermectin, believe it or not, we have some tolerance to Ivermectin now. But, you know, microorganisms, if you can grow a whole new generation in 24 hours, you can become tolerant to things pretty quickly. Ivermectin has been really hard on breaking down manure. And we've known that for a long time. I mean, you see what happens to the flies and the larvae in there. that's a bit of, I mean, in Kentucky, they were the first ones to report this, was that they were just, all the manure was piling up. And it wasn't breaking down like it used to because of the ivermectin. Nothing wrong with chemicals when we need them, but, you know, it's like, well, if a little bit works, maybe a little bit more works, and maybe I can, work with this too. So I think, it's always about, remember that you have consequences. Glyphosate, I know as much as, and I know Dr. Huber very well, and glyphosate is one of those things that we have way overused. And I'm not going to tell you differently. I mean, we just abused the heck out of that chemical. And we were helped. I mean, right? Betty Crocker Farming, open the box, add chemicals, off we go. Roundup Ready canola, Roundup Ready corn, Roundup Ready this, and it's all like no-brainer grow. You don't have to worry about the weeds. You don't have to do anything else because I got this chemical. I can do everything with it. And so we way used it, and we're totally, and a lot of farmers are really intolerant of having anything green on the ground. I mean, you have black soil, man. You got to get over it. Get over the fact you need bare soil between your rows. You don't. Even in the orchards. I talked to orchardists in California, and they're like, I need that bare soil. I said, why? Well, the grasses compete with my trees. I said, how? Well, it does. I know it does. I'm like, well, really? Like, show me. And they're like, well, but Grandpa said, and I said, I get that. And no disrespect to Grandpa, because Grandpa did the best he could, and so did your dad, and so are you. I come from ecology and plant physiology and biochemistry. I ask all the dumb questions because I'm thinking about it as a scientist and not as a farmer. So I didn't have any of that baggage. I didn't know any of that. It was like, I didn't know what Grandpa did because my grandpa, well, my dad was a hockey player, and my grandpa, did other things. So, I mean, I'm like, I don't know anything about this. So I would ask all these dumb questions, and people go, because? And I said, well, my kids, they ask, if they say because, that is not an answer. And so I don't want you to do because. I mean, I think that glyphosate has its place, and if I have biologically active soils, I can tell you for sure it gets eaten up. In fact, some of the problem we have with herbicides right now is they do get eaten up by the soil. If you're not overusing phosphorus and you need to use glyphosate, glyphosate. Glyphosate is an excellent source of phosphorus for microorganisms. And if you haven't overused phosphorus, it'll break that stuff down like nothing, and it actually won't work anymore. But don't overuse it. Don't be using around waterways. I mean, I see people doing the dumbest stuff. And it's like, did you think about what was going to happen to your kids? I mean, I remember being in a corn growers meeting in Missouri corn growers meeting, and they were talking about mercury and how that was going to change a few cents if they upgraded some of their electrical system. It was going to stop mercury gas from being emitted into the atmosphere. And they were arguing over a few cents extra to not have mercury in their air. And they were actually going to vote against it. And I just stopped and said, are you out of your minds? Do you care about future generations? Mercury is completely toxic. Do you all want to be a whole bunch of morons in the next generation? Because that's where you're going. And they're like, what? I said, it's a few cents. You're stepping over your health and the health of your children for a few cents. That's like stepping over $1 to pick up a penny. Come on, let's think about this in scientific terms. So somebody asked me, I don't sell products, and there is a reason for that, is because then I can be more unbiased. And I do want you to make decisions on your farm and your ranch based on science. I do. I really want you to make an informed decision. So do your due diligence. Don't just look at the pictures. I know it's easy to do. I used to like looking at comics too when I was a kid. And I still do. But don't just look at the pictures. Really do your due diligence. And there's a lot of resources out there. Don't just go to social media, because everything on Facebook is not true. But but have a look. Have a look around. Talk to your neighbors. Form peer groups. Talk to each other and share shared the burden of looking at some of these things. Thank you so much. I know that was my last question.
Let's give Jill one more round of applause. We really appreciate you coming out. This has been great. Before we do go to our next sessions, just a few quick things. So Emily mentioned that CIG grant that we got as the Utah Soil Health Partnership. So that is something, there is an application process. If you go to utahsoilhealth.org, you want to, if you're interested in participating in that program, there's a simple application there to just give us some information to say, yes, you're interested and want to be considered for one of the demonstration sites. So we're going to be setting up 16 demonstration sites throughout the state on farm trials with that program. So please check that out. Jay Olson is here with the UDAF water optimization program as well, which is a big thing. He wanted to just make you guys aware of a few things before we switch classes.
Yeah, appreciate the time. Like Tony said, Jay Olson, Department of Ag and Food. Hopefully everybody is aware that there is a water optimization program at the Department of Ag, and if you're not, you are now. And you can find all the information on the department's website on the front page, right above the banner. It says water optimization, and just a little bit of information on that. It's we've had 2 grant cycles. This is the 3rd one, and this grant cycle, they've given us $20 million to put towards ag water optimization. So that's a sizable amount of money. There's a $500,000 cap. It's a 50-50 grant program. So you've got to come up with the other 50%. But that can come from BOR, Water Smart, Natural Resource, you know, EQIP money and so forth. The application deadline is April 15th, and you have to document water savings. The more water savings you can document and where that water is going to be, the higher you will rank. You have to put a meter in, to meter everything. That's a requirement, real-time metering, because you've got to turn in a report every year so we can document that water savings. And I know everybody in here will be excited that they want that water to stay back in the stream if possible, but you know, you got to document that water savings. We have the potential to get another 50 million after the session. So think about what you want to do on your farms, irrigation companies, start planning, and like I said, the application is online at the department. All the information's there, my contact's there. So if you got questions, let me know. Thank you.
Okay, everybody, appreciate that. Let's, if you can, pick the next room you're headed to. We're gonna have our next sessions, and then there will be a break after that.